Hello everyone. Today I'll be discussing about some of the questions from Mudit Khanna, All India NEET PG MEE patterns. So this is a volume 1 which is from year 2020 to 2016. So right now I am doing some biochemistry question. So let's begin with question number 30, as I have already discussed about question number 29. Right now I'll be talking about question number 30, which is the following statement regarding structure of the protein is true. So A, primary, secondary, tertiary structure are destroyed by denaturation. B, three-dimensional structure of polypeptide is called secondary structure. C, secondary structure is stabilized by disulfide bridge. D, secondary and tertiary structures depends on interaction between amino acid. So looking at all these options, I think D is the correct answer, because as we can see, primary, secondary, tertiary structure are destroyed by denaturation. This is false, because primary structure cannot be destroyed by denaturations, although secondary and tertiary can be destroyed but not primary. Okay, in B options, three-dimensional structure of polypeptide is called secondary. This is also wrong, because it is a tertiary structure. Three-dimensional structure of polypeptide is called tertiary structure. And C option, you can see secondary structure is stabilized by disulfide bridge. This is also wrong. It is stabilized by hydrogen bond. So D, answer D is the correct answer. Secondary tertiary structure depends on interaction between the amino acids. So we can see the explanations also. Let us go to the explanation. As here there is explanation. Here what it has written is secondary and tertiary structure depends on interactions between amino acids. Alright, so this is the answer given in the questions. Secondary structures depends on simple interaction, hydrogen bond, between backbone atoms of primary chain without involvement of side chain R group, whereas tertiary structures depends on more complex interaction like hydrogen ionic covalent disulfide bonds. Okay, so along the involvement of side chains atom or R group. So this is the correct answer. Denaturation of protein does not affected by primary structure. Does not affect primary structure. The secondary, tertiary, quaternary structures are destroyed by denaturations, change in pH or temperatures. Denaturation of protein does not affect the primary structures. Denaturation involves secondary up to quaternary structures of protein and does not involve the protein primary structures. So this is also false. Alright, so three-dimensional structure of polypeptide is called tertiary structures. The overall three-dimensional structures, 3D conformation of polypeptide chain is called tertiary structure. This tertiary structure is required to perform a chemical physical tasks. Alright, secondary structure represents only a three-dimensional pattern of localized segment of polypeptide chains. What it has said is secondary structure represents only a three-dimensional pattern of a localized segment of polypeptide chain. The 3D pattern of secondary structure, a helix or beta pleats, is not representative of 3D structure of the polypeptide chain or protein and is insufficient to perform a particular chemical or physical task. Alright, so we can understand from this statement also, the secondary structure represents only a three-dimensional pattern of a localized segment of polypeptide chain. The 3D pattern of secondary structures, a helix or beta pleats, is not representative of 3D structures of polypeptide chain or protein and is insufficient to perform a particular chemical or physical task. So this is the answer. So the next one option which was given in the question for that question number 30 was secondary structures are stabilized by hydrogen bond, not by disulfide bond. Alright, the disulfide bond are found in tertiary. These disulfide bond are covalent linkage between the sulfur containing side chains of cysteine are more stronger than other type of bond that contribute to tertiary structures. They act like molecular safety pin, keeping parts of polypeptides rigidly attached to one another. So this is the answer. Alright, so what it has said just now is very important in other questions also. So the furthermore there are some explanation of these questions. Let us go in deeply. What is the protein structures, what are the primary, secondary, tertiary, quaternary structures are there. The protein structure is normally described at 4 levels of organizations. It has mentioned about this also. Primary, what is the primary? It is the simplest level of protein structure. It is simply the sequence of amino acid in the polypeptide chain held together by peptide bond. So peptide bonds holds together all the amino acids in a polypeptide chain. Alright. So primary structure is insufficient to perform particular chemical or physical tasks. Denaturation of protein does not affect the primary structure. Denaturation involves secondary up to quaternary structure of a protein and does not involve the protein primary structure. So this is the primary structure of protein, and this is primary structure is sequence of chain of amino acid. Alright, so this is the chain, and these are the amino acid is small dotted, and these are connected by peptide bond. So what is secondary structure? Secondary structure is a produced from localized folding or coiling of parts of peptide chain in different spatial arrangement. 3D pattern of localized segment of polypeptide. The most common type of secondary structure are alpha helix and a beta pleated sheet. A single polypeptide may exhibit all types of secondary structures. So if it is a single polypeptide chain, then that is then exhibits all types of secondary structures. Alright, that exhibits. This occurs due to hydrogen bond formed between atoms of primary chain backbone. The backbone just refers to the polypeptide chain apart from R group. Thus, secondary structure does not involve R group atoms. Secondary structure is also insufficient to perform particular chemical or physical tasks. Both primary and secondary are insufficient to perform chemical or physical tasks. So you can see the structure. Secondary structure formed due to local folding of the polypeptide chains due to hydrogen bond. So this is alpha helix, this is beta pleated sheet. So these are the secondary structures. It is more complex than that of primary structure, and these alpha helix and beta pleated are the most common types of secondary structure. Alright, so this single polypeptide exhibit all types of secondary structures. So let's move on towards the tertiary structures. So you can see the pictures of the tertiary structures over here. It is a 3D folding of a secondary structure due to side chain interaction. So here R group interaction will be present, whereas in secondary R group interaction won't be there. So let's start from the beginning. The polypeptide chains with secondary structure are further folded, superfolded and twisted to form tertiary structures. So if thus if there is a secondary structure is formed by localized folding or coiling of parts of polypeptide chain in different spatial arrangement, that is secondary. And if the polypeptide chains which are secondary in structures are further folded, superfolded, twisted to form a tertiary structures. The tertiary structure is primarily due to interaction between the R group of amino acids that makes up the protein. So there is an interaction between the R group of the amino acid that makes up the protein. So interaction that contribute to tertiary structure include non-covalent bond, that are hydrogen bond, ionic bond, dipole interactions, London dispersion force, and covalent bond, that is disulfide bond. Covalent bond is a disulfide bond. So these interactions between all these non-covalent and covalent bonds, this helps to form the tertiary structures. The overall three-dimensional structures of polypeptide is called tertiary structures. 3D structures are called tertiary structures. So this tertiary structure is sufficient to perform the particular chemical or physical tasks, but primary and secondary won't be able to perform such kind of chemical or physical tasks. Let's go to the quaternary structure. Many proteins are made of single polypeptide chain and have only 3 levels of structures, that is tertiary structures. However, some proteins are made of multiple polypeptide chains, also known as subunits. When multiple polypeptide subunits are present, the spatial relationship between these subunits is referred to as a quaternary structure. When multiple polypeptide subunits are present, the spatial relationship between these subunits is referred to as a quaternary. Example of important protein with quaternary structures include creatine kinase is dimer, 2 monomer spatially arranged. So creatine kinase is a dimer and 2 monomer spatially arranged. Hemoglobin is a tetramer made up of 2 alpha chains and 2 beta chains. Immunoglobulin G is tetramer made up of 2 heavy chains and 2 light chains. Lactate dehydrogenase is a tetramer. So these are the complex proteins, and you can see the structures. Quaternary structure is when multiple polypeptide subunits are in spatial relationship. These are in spatial relationship. So I want to conclude on this video. That's question number 30 that I have just completed from the Biochem, from the Mudit Khanna book. Alright, so hopefully there is some clear in the conceptions between the primary, secondary, tertiary and quaternary structures, and I think it will be easier to solve the questions in future if it comes in exam. Thank you very much.